Okay, so the topic is using convolutional neural network to analyze bacteriophages DNA.
Okay, hi, I'm Michael. This is my colleague Bacteriophage, and today we're going to talk about using convolutional neural networks to analyze bacteriophages DNA. But before we start, some of you might be wondering what the bacteriophages even are. So how many of you heard about the bacteriophages? Okay, quite a bit of you. So the bacteriophages are viruses that target bacteria. They are these tiny microbes that target specific strains of bacteria in your system. They kind of work like these tiny syringes. So what makes them interesting is that the antibiotics usually target all strains of the bacteria, while bacteriophages can target specific strains, which makes them really useful. As far as the industry use goes, they are used in diagnostics to, for example, detect if a certain bacteria strain is present in a sample. They are also used in medicine as an alternative to the antibiotics. So antibiotics work kind of like bombs. They kill both good and bad bacteria in your system, which isn't ideal. Bacteriophages can be designed to target the specific strain that causes your actual disease, which makes them much more safer. So because of that, they are also widely used in agriculture. And the overuse of antibiotics in agriculture causes the emergence of the so-called superbugs, which are drug-resistant bacteria. And of course, we don't want drug-resistant diseases out there. So again, bacteriophages are a much more safer and natural alternative to that. But of course, we cannot just use any bacteriophage strain before we make the so-called phage cocktails. We first have to analyze the strain, which might take up to weeks of laboratory work, to check if they meet all of the criteria. So one of the most important criteria is how they reproduce. Based on the reproduction cycle, we can detect two distinct types of bacteriophages, lytic and lysogenic. So the lytic bacteriophages kill the bacteria right away. At first, they attach themselves to the bacterium, then they inject their DNA and start eating the bacteria from inside out. Then they synthesize the proteins and start assembling new bacteriophage organisms, and finally release, killing the bacteria. So yeah, nature is pretty brutal. The second cycle type is lysogenic. So it starts pretty similarly. At first, the bacteriophage attaches itself to the bacterium and then injects its DNA. But instead of eating right away, it just kind of waits in this dormant stage where it waits for the bacterium to replicate. And when the bacterium replicates, it not only replicates its own DNA, but also the DNA of the bacteriophage, allowing it to reproduce with it. And then when certain conditions are met, the bacteriophage enters lytic cycle, killing the bacteria altogether. So these two cycles might seem pretty similar, but the key differences are vast. So the lytic bacteriophages kill the bacteria right away, and there's no problem. But the lysogenic bacteriophages can cut and move parts of DNA between the bacteria, which might cause certain mutations. And again, we don't want new diseases caused by mutated bacteria out there. So before we use a bacteriophage strain commercially, we have to make sure that it's lytic. So a little bit about the origin of the project. We recently went to the BioHack Hackathon organized in Łódź with a team of programmers and data scientists. It's a small competition that you have to create an application in about 24 hours on one of the given topics. So the topic that we chose was proposed by the Proteon Pharmaceuticals company. And the goal of the task was to create an automated classifier that detects whether the bacteriophage is lytic or lysogenic. So this company deals with bacteriophages daily, and they spend weeks analyzing their genomes. So the task was to automate that process and leave the biologists with less work. We actually managed to win this competition, and so I'll tell you how. But first, let's focus on the data. Throughout the competition, we managed to crawl about 2,000 bacteriophage genomes, which means we had about 2,000 separate organisms that we used as the training set. And I mean, the data set was pretty much balanced. There were a little bit more lysogenic bacteriophages, but not enough to cause any significant problems. So the real problem was the size of a single sample. Each genome had up to 200,000 nucleotides, which is a lot of features, much more than many classic machine learning algorithms can handle in reasonable time. But also, since DNA is sequential, most algorithms aren't even prepared to handle that kind of data. And when we talk about DNA as data, in this case, we should think about sequence of nucleotides. There are 4 possible nucleotides. So basically, we work with strings of A's, T's, C's, and G's. And when you look for publications that will deal with DNA as input data, they usually focus on a single species. So if you take any 2 humans, the DNA is pretty much the same. There are a few nucleotide differences that you can treat as categorical features, for example, but most of the genome is pretty much the same. In case of bacteriophages, we actually had to work with multiple strains, which means we had to work with the whole genomes. We had to analyze the whole DNA sequence rather than using some derived features. And as I've said, the genome could be huge. It could contain up to 200,000 nucleotides, which is pretty much very problematic. So in order to fix that problem, we used convolutional neural networks. Convolutional neural networks were originally designed to handle image data, but really, as long as you prepare the input correctly, they can pretty much handle any kind of sequential data there is. So what makes convolutional neural networks really good at this is that they create this sort of reusable kernels or filters that look for specific patterns throughout the whole input rather than trying to analyze the whole input at once. So in case of DNA, this is really important because if a certain bacteriophage can synthesize a protein that is unique to lytic or lysogenic bacteriophages, then it doesn't really matter if the protein is encoded at the beginning or at the end of the sequence. We just care that it can synthesize it, that it's out there. So you cannot really use any ready image processing architecture. Of course, you had to prepare it for the DNA. So let me give you a high-level overview of our model that we've built. So as input data, we use nucleotide sequences, so basically strings of A's, T's, C's, and G's. Then we perform simple one-hot encoding vectorization. So for each nucleotide, we assigned a unique vector that represents it. So for example, A was represented by 1, 0, 0, 0, T was represented by 0, 1, 0, 0, and so on. This allowed us to convert from string input data type into series of vectors or matrices that represent the whole genome. So after the data was vectorized, we passed it into the first convolutional layer. So this layer was designed to detect codons. Codons are triplets of nucleotides that encode certain amino acids. So the goal of this layer was to convert from this sort of sparse representation of the nucleotides into dense representations of amino acids. Then we passed the data into further convolutional layers. So these layers detected higher level features. So for example, the first one could detect amino acids, the next ones could detect proteins, and the last one could detect sets of proteins throughout the whole genome. Then we converted data into fixed size vectors, which allowed us to use standard dense fully connected layers. So these layers processed data further and finally connected with the output layer, which gave us probabilities for each class. Now, this might seem like any regular image processing pipeline except for the input type, but we did have to apply some tricks in order to make it work. So first we had to use much wider convolutional kernels. So in most architectures you use these kind of small filters that process like 3 or 5 pixels at once, but in the case of DNA it didn't really work. Before we made sure that the last convolutional layers could actually see the whole genome, we weren't able to build any sensible models. So they all returned 1 or 0, which isn't ideal. So before we made sure that the model can actually see the whole genome, we weren't able to build any useful models. The second simple trick was using skip layer connections. So skip layer connections is this neat trick that was used in the recent architecture, and it's very simple. So instead of placing your layers sequentially one after another, you allow for some arbitrary connections between the layers, which in turn allows the layers to handle both low and high level data, so it doesn't forget the original input. So it didn't exactly improve our scores on the validation set by much, but it did improve our scores on the testing set on unseen data, which makes us believe it's very helpful for the generalization of the network. And the last thing we did was using some actual expert knowledge. So it might seem like you can just throw the data at the model and hope that it finds all the features by itself. And we kind of did that. But also using some domain knowledge to improve the model might be helpful. So in our case, when we used the first convolution layer, which was specifically designed to handle amino acids, it did improve our scores on the testing sets. And of course, there are other issues, like the DNA was so huge that it didn't fit in the memory, so we had to use really small batches. But let's not bore you with that, and let's go to the accuracy. So as far as the results go, we managed to achieve about 97 accuracy on the validation set and 100 accuracy on the testing set. And before you say that this 100 sounds suspicious, because it kind of does, it's only because during the competition we received only about 10 or so samples that were fully labeled, so it wasn't really hard to achieve. But we still expect the accuracy to be over 90% on unseen data. And I mean, this is pretty much enough for the biologists to speed up their workflow. We obviously can't replace the biologists just yet. But for now, we can tell them that certain bacteriophages are more interesting than the others. So for example, if we can tell that this bacteriophage is lysogenic and you're wasting your time analyzing it, and these ones are lytic, so you should focus on them, then it helps with their work. But obviously, the biologists won't trust us before they can understand what we're doing, so we had to focus on the interpretability from day one. So at first we applied LIME, which stands for Local Interpretable Model Agnostic Explanations. So LIME builds a second simpler model based on your original classifier to check which features are actually important. It works like this. So at first it generates a variance of your original input that you're trying to understand. So for example, slightly distorted DNAs. Then it passes the distorted versions of the input into your original model to see how it reacts to the changes. And after it sees how it reacts, it can build a second model that tries to mimic your original one and check which features are actually important. And the second model is much more simple and it can be easily interpretable. So in case of LIME, it works really well for like text processing, but in case of DNA, it didn't really work at all. First of all, it took ages to build, and I mean like we had to wait minutes for a single input to complete, and it isn't really production grade. And more importantly, the results were not reproducible. So if you've built like 3 separate LIME models, you would get 3 completely different sets of features that are important, which wasn't really good in our case. So instead we use class activation maps. Class activation maps is this neat algorithm that allows you to visualize activations of your each layer. So basically you can see what the neurons are actually looking at in relation to the class. For each output class of your model, you can generate separate class activation maps, which is really nice because we could highlight both lytic and lysogenic features in the original input. So this really helps us with building the models, because if we can see that they focus on kind of junk DNA instead of the actual important features, we can rebuild the model and wait until it works correctly. So for example, if it highlights proteins unique to lytic or lysogenic bacteriophages, then it's good, it's great, the model works. But if it highlights kind of random DNA parts, that might mean that they're looking for specific bacteriophage strains instead of the actual important features. So we have to rebuild the model. As far as future steps go, the most obvious one is using more feature engineering. So for example, now we're using very simple vectorization method, and there are so many ways that you can represent DNA. Instead of using nucleotide sequences, we can focus on amino acid sequences. We could also extract proteins and treat them as categorical features. But what I'm most personally interested in is using DNA embeddings. So embeddings is this kind of way of representing data or certain entities in vector space. You assign a unique vector to each of the entities that you're trying to analyze. So for example, it's very popular in the NLP, where you build embeddings of words and sentences. But in case of DNA, we could build embeddings for proteins. If 2 separate proteins are similar to each other in the way they work, but they are structurally different, their embeddings or their representations should be pretty much similar to each other, which tells the model that even though the proteins look different, they do the same thing. And when you build the embeddings, you only have to do it once. So the biologist can confirm that, yeah, this looks okay. And then we can just pass it into multiple models and rebuild them and so on. But in case of full models that try to extract this kind of embeddings on their own, you have to validate each model more thoroughly. So it takes more time. This is something that I want to focus my PhD studies on. So obviously we can also try using other architecture and other algorithms. So during the competition we only had time to try out a few architectures, but now that we can focus on the problem, we can try completely different approaches. And also we're building a full-fledged application with protein pharmaceuticals for all things bacteriophage. So for example, it will have a full bacteriophage database. You can upload your bacteriophages, you could classify them, see the results, highlight the features, and so on. This is something that we're currently working on, so yeah, wish us luck. So this is all from me. Thank you for listening, thank you for having me, and if you got any questions, don't hesitate to ask.
I have a question about your background. Did you have something in touch with biology before?
I knew that DNA exists, but that's pretty much it. Yeah.
You've told us about your model that you've inspired by ResNet and so on. Do you have your own model, or did you copy some existing models?
Yeah, it was built from scratch, but with keras, so there are a lot of pre-implemented blocks that you can kind of put on together.
Yeah, sure. And have you taken a look on the Squeeze and Housed? It's the latest ImageNet winner.
No.
Okay, and last question, sorry. How much do you have a data in dataset, your training dataset?
About 2,000 bacteriophages.
Okay,
but
nice.
The size of the dataset is not that much important, but the size of a single sample is, because we couldn't build really huge models because they wouldn't fit in the GPU. So that was the most problematic part about this.
Thank you.
We couldn't really use very complex architectures because it just wouldn't fit in the GPU.
Thank you.
Okay, thanks.